Assalamu alaikum. I am Dr. Sani Asif. Today we will discuss about the histology of the connective tissue. What is connective tissue? This is a supporting tissue which support all the cells and the components in the body. Its contents are the introduction. We will discuss its introduction. Then we will classify the connective tissue. Then we will discuss the functions of the connective tissue. Then what are the components of connective tissue and types? These are all our learning objectives which we will discuss in this lecture. First of all, introduction. Most widespread and abundant type of tissue in the human body is connective tissue. Major constituent is the extracellular matrix, which is composed of fibers, ground substance, and tissue fluid. Embedded within the extracellular matrix are the connective tissue cells. So mainly the connective tissue has three components, which are cells, fibers, and ground substance. It forms a vast and continuous compartment throughout the body, bounded by basal lamina of epithelia and by basal lamina of muscle, nerves, and vascular endothelium. Now, about the classification of the connective tissue. Classification is based on the composition and organization of cellular and extracellular components and on special functions. It is mainly classified into three types: connective tissue proper, specialized connective tissue, and embryonic connective tissue. Now we will discuss them one by one. First of all, connective tissue proper. It is further divided into the loose connective tissue and dense connective tissue. Loose connective tissue, as the name indicates, it has less number of fibers, and in case of dense connective tissue, there will be more number of fibers. Dense connective tissue is further divided into the regularly arranged dense connective tissue and irregularly arranged connective tissue fibers. Then the specialized connective tissue. Specialized connective tissue have adipose connective tissue, reticular connective tissue, and elastic connective tissue. These are the three types of specialized connective tissue. Firstly, we discuss the connective tissue proper, then specialized connective tissue, and now the embryonal connective tissue, which is divided into the mesenchymal connective tissue and mucous connective tissue. What are the functions of connective tissue? It is very much abundant. Then what are its functions? It forms the capsules that surrounds the organs of the body and the internal architecture. It makes up the tendons, ligaments, and areolar tissue that fills the spaces between the tissues. Bone, cartilage, and adipose tissue are specialized type of connective tissue that support the soft tissue of the body and store fat. It has the role in defending the organism due to the phagocytic and immunocomponent cells. Phagocytic cells engulf inert particles and microorganisms that enter the body. Specific proteins called the antibodies are produced by the plasma cells in the connective tissue. Plasma cells are one of the cells which are present in the connective tissue, so they produce the antibodies. Connective tissue provides physical barrier. It plays a role in the cell nutrition and serves as a medium through which nutrients and metabolic waste are exchanged between cells and their blood supply. These were the few functions. Now we will discuss about the main components, basic components of the connective tissue. What are these? These are ground substance, fibers, and cells. Firstly, we will discuss the ground substance. Ground substance is a very major component of the connective tissue. A complex mixture of glycoproteins and proteoglycans make up the ground substance. They participate in binding cells to the fibers of the connective tissues. It is colorless and transparent. It fills the space between the cells and the fibers. and its consistency is viscous. It acts as both a lubricant and barrier to the penetration of foreign particles. Its nature is granular and its consistency varies from fluid to gel. Glycoproteins, which are the main component of the ground substance of the connective tissue, these are proteins that contain oligosaccharide chains covalently attached to the polypeptide side chains. The carbohydrate is attached to the protein in a co-translational or post-translational modification. The major types of adhesive glycoproteins are the fibronectin, laminin, and intectin. Fibronectin, which is a glycoprotein synthesized by the fibroblast and some epithelial cells, it binds with the collagen. It connects collagen fibers to the cells of the connective tissue. Next is the fibrillin. It forms the elastic fibers in connective tissue and responsible for adhesion of different extracellular components to one another. Then last is the glycoprotein is the laminin. It is present in the basement membrane. Laminin helps in adhesion of epithelial cells to the basal lamina. Intectin, it is an adhesive glycoprotein and it is seen in the embryonic tissue and plays a role in the cell migration. There are also other glycoproteins which are chondronectin and osteopontin. Chondronectin and osteonectin are similar to the fibronectin. Chondronectin has binding sites for type 2 collagen, chondrite and sulfates, hyaluronic acid and integrins of chondroblast and chondrocytes, while osteonectin possesses domains for the type 1 collagen, proteoglycans and integrins of osteoblast and osteocytes. This is the table which is showing the tissue distribution and functions of these glycoproteins. Means fibronectin, which is mainly widely distributed in the extracellular structures, cell surfaces and fibroblast, and their function is the cell adhesion mainly. Then laminin, its tissue distribution is the basal lamina and the external lamina of the muscle, and its function is to bind to the epithelial and muscle cells. Then intectin, which is present in the basal lamina again and binds to the laminin and type 4 collagen. Then these other are also very important. Chondronectin, which is present in the cartilage, and osteopontin, as the name indicates, is present in the bone. Other substances in the ground substance are the Glycosaminoglycans or the mucopolysaccharides. These are the linear polysaccharides formed by the repeating disaccharide units. They are composed of a uronic acid and a hexosamine. Hexosamine can be glucosamine or galactosamine, while uronic acid can be glucuronic acid or iduronic acid. Linear chains are bound covalently to a protein core. Glycosaminoglycans are long, inflexible, unbranched polysaccharides composed of chains of repeating disaccharide units. In cartilage, these are bound to the hyaluronic acid chain. Because of the abundance of the hydroxyl, carboxyl, and sulfate groups, the proteoglycans are hydrophilic. Hydrophilic means water-loving, and they act as the polyanion. Then these are some glycosaminoglycans. Their tissue distribution and functions are there. Number 1 is the hyaluronic acid, which is widely distributed, found in various amounts in all tissues and fluids in the adults: loose connective tissue, skin, umbilical cord, vitreous, synovial fluid, and the cartilage. And its function is to resist compressive forces in tissue and joints and space filler during embryonic development, and also facilitate the cell migration. Next one is the chondrite and sulfate, which is located in the hyaline and the elastic cartilage and the bone, and it provides the mechanical support and forms large aggregations with the hyaluronic acid. Then is the dermatone sulfate, which is present in the dermis, tendons, ligaments, heart valves, and organ capsules, and they bind to the type 1 collagen fibrils. Then the keratin sulfate, which is present in the bone, cartilage, cornea, and it provides the mechanical support. Then is the heparin sulfate, which are present in the fibroblast and epithelial surface, basal and external lamina, and its function is the cell adhesion, and it binds fibroblast growth factors, structural and filtering functions in the basal lamina. Proteoglycans, when sulfated glycosaminoglycans form covalent bonds with a protein core, they form a family of macromolecules known as the proteoglycans. They look like a bottle brush with the protein core resembling the wire stem and various sulfated glycosaminoglycans projecting from its surface in three-dimensional space, as do the bristles of the brush. They are just like that. As you can see in this diagram, this is the picture which is showing the proteoglycan. Here the proteoglycan consists of a core protein. Here you can see this is the core protein and consists of a that binds the glycosaminoglycans. These are the sulfated glycosaminoglycans. These are attached to the core protein. And a link protein that binds hyaluronic acid. Again, a link protein is here. This one is a link protein that will bind to the hyaluronic acid. This one is the hyaluronic acid. So till now we have discussed the first component of the connective tissue, which is the ground substance. Now, what are the functions of the ground substance? They transport metabolites to and from the vascular channels. They maintain the electrolyte balance, fills up the space between cells and fibers of the connective tissue. They act as a barrier to penetration of foreign particles into the tissues. Connective tissue fibers are long, slender protein polymers. We now discuss about what are the different types of the connective tissue fibers. As I told you in my initial slides that connective tissue has mainly three components: ground substance, fibers, and cells. Up till now we have discussed about the ground substance. We have discussed about its components, which are proteoglycans, glycoproteins, etc. And we have discussed its functions. Now we are coming to the second component of the connective tissue, which are the connective tissue fibers. There are three types of connective tissue fibers which are present in our body, which are collagen fibers, reticular fibers, and elastic fibers. First of all, we will discuss the collagen fibers. Collagen protein, this is the most abundant protein in our body. So, the most abundant protein in mammals is the collagen, and it accounts for the 25 to 30% of the total protein content. Main fibrous component of the skin, bone, tendon, cartilage, and teeth. All these components or the structures have the collagen fibers. It comprises about 90% of the organic matrix of the bone. That is why the bone has a strong and has increased strength due to the presence of the 90% organic matrix in the form of collagen fibers. Word collagen, colla, comes from the Greek meaning glue producer. When collagen is heated in water, it gradually breaks down to produce soluble derived protein, that is gelatin or animal glue. Here you can see when you will heat it in water how it get convert, and when you cool it become in the gelatin formation. The collagen fibers are made up of collagen, as you can see here, and they have a tensile strength, an important structural component in tissues such as the periodontal ligament and muscle tendons, in which the mechanical forces need to be transmitted without loss. Now, what is the structure of the collagen fibers? Collagen subunit or tropocollagen is a rod about 300 nanometer long and 1.5 nanometer in diameter. It is composed of three polypeptide alpha chains which are coiled around each other to form the triple helix configuration, which is stabilized by numerous H2 bonds. Individual polypeptide chains contain approximately 1000 amino acid residues, and alpha chains are left-handed helices that wrap around each other into a right-handed rope like triple helical rod. This diagram you can see here. This is the tropocollagen helix. Then we will discuss the biosynthesis of collagen. The collagen is involved in tissue differentiation, growth and remodeling. Young tissue has high rate of collagen synthesis. As the tissue matures in adults, synthesis continues as a part of normal tissue turnover. They have the highest rate of collagen turnover are observed in weight-bearing bones, lungs and periodontal tissues. Collagen synthesis is elevated under conditions like remodeling and replacement of tissues and during tissue repair. Elevated rates in pathological conditions such as fibrosis in lungs and liver. Now comes to the synthesis of the collagen. Firstly, there are the synthesis of pro-alpha chains. These are in turn undergo hydroxylation of proline and lysine residues. After that, the hydroxylation, there will be the subsequent glycosylation of these hydroxylysine in ribosomes. After that, there will be the assembly of pro-alpha chains into the triple helix. Packaging of the procollagen by the Golgi into the secretory vesicles. Then these transport of procollagen containing vesicles along cytoplasmic microtubules to the cell surface. Then there will be the cleavage of registration peptides to form tropocollagen molecules, and then the assembly of tropocollagen molecules into the microfibrils, and side by side cross-linking of the collagen fibrils to form the collagen fibers. Vitamin C has a very important role in the hydroxylation process in the synthesis of the collagen. Cross-linking of the collagen play a major role in structure and function. Most important cross-links in collagen are derived from specific lysine and hydroxylysine. If there is decrease in the cross-links, it will reduce the tensile strength of the collagen and contributes tissue fragility. With increasing age, collagen becomes more and more cross-linked. Now we will discuss about the clinical significance of the collagen. Collagen are the most abundant proteins, and alteration in the collagen structure resulting from abnormal genes or abnormal processing result in numerous diseases. The most common are the osteogenesis imperfecta, scurvy, Ehlers-Danlos syndrome, Alport syndrome, epidermolysis bullosa, Strickler syndrome, lupus erythematosus, and scleroderma. This is the diagram of the patient with the Ehlers-Danlos syndrome. Here you can see there will be abnormal ability to elevate the right toe. In this diagram, the girl with the Ehlers-Danlos syndrome, the dorsiflexion of all fingers is easy and absolutely painless. And in this diagram, you can see patients with the Ehlers-Danlos syndrome have the joint hypermobility is less intense than with the other conditions. This is the diagram showing the patient with the SLE, which is the systemic lupus erythematosus. Here you can see the classical butterfly, this one. So up till now we have discussed about what is connective tissue and its components, which are the ground substance, fibers and the cells. Also we discuss about its classification, and we discuss in the component the ground substance in the detail. Then we discuss about its fibers, in which we have discussed about the collagen fibers, the most abundance of the collagen protein, its cross linkage and its importance, then its biosynthesis and the clinical correlations. Now we will discuss the second type of fibers which are present in the connective tissue, which are reticular fibers. These are extremely thin with a diameter between 0.5 to 2 micron meter. They form an extensive network, not visible in the HNE, you will say the hematoxylin and eosin preparations. They stain black by impregnation with the silver salts, and they are periodic acid shift positive. This is a reagent of the staining where they give the magenta color. They contain 6 to 12% hexose and composed mainly of the collagen type 3 in association with other types of collagen, glycoproteins and proteoglycans. They are formed by loosely packed thin fibrils bound together by abundant small interfibrillar bridges composed of glycoproteins and proteoglycans. They are abundant in the smooth muscle, endoneurium and framework of the hemopoietic organs. All the lymphatic organs you will discuss, the lymphoid system, they have the reticular fibers, and they constitute a network around the cells of the parenchymal organs. Small diameter and loose disposition of reticular fibers create a flexible network in organs that are subjected to changes in the form or volume, such as arteries, spleen, liver, uterus, and intestinal muscle layers. Elastic fiber system. Structure of the elastic fiber system developed through three successive stages. First stage consists of the bundle of 10 nanometer microfibrils composed of various glycoproteins, including one with a large molecule called fibrillin. These oxytocin fibers can be found in the zonule fibers of the eye and the dermis. In case of the second stage, an irregular deposition of the protein elastin appears between the oxytocin fibers, forming the Laminin fibers. These structures are found around sweat glands and the dermis. Third stage in the formation of the elastic fiber system is where the elastin gradually accumulates until it occupies the center of the fiber bundles, which are further surrounded by a thin sheet of microfibrils. These are the elastic fibers, the most numerous component of the elastic fiber system. Elastic fibers, which are rich in protein elastin, they stretch easily in response to tension. I hope you have understood well the ground substance and the fibers. In the next lecture, we will discuss about the cells. Thank you.